So hello, welcome to the Cambridge Clinical Vision Laboratory, and today I'm just going to talk to you a little bit about visual evoked potentials, which is one of our really important tests for quantifying the effects of remyelination promoting drugs. So visual evoked potentials have been around for many years, and actually this was previously one of the key diagnostic tests for MS. And the technique is essentially to present a stimulus, a visual stimulus, to a patient. And normally that looks something like a chessboard that flashes about twice a second, and we record from the scalp the electrical signal that's generated in the brain in response to that visual stimulus. And in conditions like multiple sclerosis, where you lose the lining of the nerve fibres conduct more slowly, and as a result it takes longer for the electrical signal to reach the back of the brain. And that's why it's so valuable, this test is so valuable to us, to capture the effects of remyelinating drugs. Because in response to remyelination, the time taken for that visual stimulus to yield an electrical response in the brain should be shorter. So that's one of the things that we mean when we talk about the latency of the visual evoked potential, how long it takes that signal to reach the back of the brain. Because visual evoked potentials are such a powerful and sensitive way of capturing remyelination, this has been our primary outcome measure for our most recent trial, CCMR Two, which is testing metformin and clemastine in people with relapsing-remitting MS. And because it's our primary outcome measure, we needed to ensure that every single person coming onto the trial had measurable demyelination, or slowing of the visual nerves, in order to get onto the trial. Because in order to measure the repair or recovery, there had to be a degree of abnormality there in the first place. And actually, that's one of the key barriers we faced with this trial, because we were anticipating about 60% of people would have a measurable abnormality, but actually it's been closer to 40%. And that is, in many ways, a reflection of how effective our MS treatments are. Now, fewer people just happen to have measurable demyelination in the visual nerves. So as a result, we've been able to use visual evoked potentials as our outcome measure for the trial. And what we are doing is using this test over the course of 6 months to see if there's a recovery of visual evoked potential latency in those treated compared to those treated with placebo. So we can compare the effects on those two groups. If a drug promotes remyelination, we would expect the visual evoked potential to improve in those treated with the drugs compared to those treated with placebo. In those treated with placebo, we would expect the time taken from the visual stimulus to the recorded signal in the brain to be the same, or maybe even to get slightly worse over time. Whereas those people treated with a remyelinating promoting drug, when we put insulation back on the nerve fibres, they conduct faster, and therefore that time gets shorter. So we would expect that improvement in those treated with the drugs compared to those treated with placebo. And that's what we're looking for when it comes to the main outcome measure of CCMR Two, to see if there's been an improvement in those visual nerves of people treated with the drugs compared to placebo.